The Ethical Aspects of Genome Editing by CRISPR Technology. First author, Christine Kuzminski, Faculty Supervisor, Kiarsh Aramesh, PhD. Genome editing is a method for modifying genetic information within the cells of a living organism. The most advanced form of this technology, called CRISPR, has the potential for treating a multitude of genetic diseases in humans. However, this technology has raised some ethical questions and concerns, including concerns about its safety, modifying human germline, its application in human embryos and fetuses, use of human embryos for such research, how to obtain an informed consent, and the concerns centered around the principles of justice and equity. This independent study explores these ethical concerns and the rationale behind how these can be addressed. As a practical example, this study focuses on the treatment of Huntington's disease. This degenerative and fatal disease is a prime candidate to be potentially cured by CRISPR technology. This study explores the specific implications of the above-mentioned ethical concerns on the future use of this technology to treat this disease. As an overview, we will go over how CRISPR-Cas9 works, what the ethical concerns are individually, and what is CRISPR's potential to eliminate diseases. So this is how CRISPR-Cas9 works. First, a cell is transfected with a DNA plasmid that expresses both the Cas9 protein and a sequence of guide RNA, which matches that of the gene interest. Then Cas9 identifies the corresponding DNA sequence on the host cell's genome and cuts both strands of DNA. Then the cells attempt to repair the break effectively silences the targeted gene by joining the cleaved DNA back together using a process called non-homologous end joining, or NHEJ. Or a faulty gene can be corrected with a replacement segment of DNA, or a new gene altogether can be introduced. If a modified piece of DNA whose flanking regions match the target sequence is also supplied, then there is a good chance that it will recombine with the host DNA when the cut is made, thus introducing a new or replacement gene. This pathway is known as homology-directed repair, HDR. Cas9 requires a simple and common sequence of base pairs called a PAM sequence to actually bind to target DNA. This feature means bacteria can prevent Cas9 from chopping up important memorized sequences of foreign DNA in their own genome by ensuring there are no PAM sequences in those regions. So there's 3 applications of CRISPR. 1, turning genes on or off, medications and or self-therapy, and farming. So we can manipulate genes within humans by turning them on or off, and medications can be infused, or in some cases self-therapy, taking blood in certain cells out of a body and manipulating them with CRISPR and then putting them back in. The third, which sometimes is overlooked, is actually in farming, both farming with animals as well as farming with crops. The application of CRISPR to food has already been done. There are companies that have already been using CRISPR to create enhanced foods to resist bacteria or viruses, to create even better tasting foods. The application on animals has also been done, an example being CRISPR mice. So what comes next? Food and livestock modification, which researchers have already created plants and mammals with edited genomes. It is hoped such technology could help boost productivity and improve food security. Gene drive. Some genes are more likely to be passed on than others. If an edit is linked to these genes, it will quickly spread through a wild population. That sounds alarming, but could help eradicate malaria carrying mosquitoes. Gene therapy, genetics disease, could be treated by introducing gene editing systems into affected cells. Researchers in the USA are trialing this to treat HIV by knocking out the gene for the specific T-cell receptor that the virus targets. Human germline, modifying human embryos, sperm, or eggs would introduce changes to the genome of future generations. Some argue that other techniques such as embryo screening can just as effectively prevent genetic disease. Then there's designer organisms and more. In future, could babies be designed with a genome of our choosing? Could amateur biologists do their own gene editing outside of regulatory systems? These are all questions and likely possible outcomes. So ethical concerns include safety, modifying human germline, application in human embryos and fetuses, use of human embryos for such research, how to obtain an informed consent, and concerns over justice and equity. So the safety concerns. CRISPR has the potential to alter cells and eliminate their cancer-fighting properties, causing more damage to genes than good. There's also the likelihood of designer babies. The elite could potentially be able to design their own offspring with higher intelligence or athleticism. This messes with natural selection far beyond what we already have. Then there's side effects and downstream effects, which we think we're measuring exactly what changes we're doing to the genes, but there's always the possibility that our technology can't pick up on other changes. The safety concern is that in this field, it's moving so quickly, and some researchers want to get into human clinical trials right away, even before the CRISPR technology paradigm has been fully validated. So when it comes to modifying the human germline, we run into an issue of gene drive. One of the biggest risks of CRISPR is what's called gene drive or genetic drive. What that means is that because you're actually manipulating genes and those genes get incorporated into the genome, basically that sits within cells. Potentially those genes can then be transferred onto other organisms. And once they're transferred onto other organisms, once they become a part of the cycle, then those genes are in the environment and will be passed on from generation to generation. So, application to human embryos and fetuses, and also their use in research. The most detailed analysis to date of CRISPR in human embryos shows that applying gene editing technology to repair a blindness causing gene early in the development of a human embryo often eliminates an entire chromosome or a large section of it. So, you're going to see a lot of chromosomal abnormalities with all of the research that has been currently done on human embryos. And from that one study, they quoted, "Our study shows that CRISPR-Cas9 is not yet ready for clinical use to correct mutations at this stage of human development." So they're hoping that their studies can be a precautionary way of holding off human clinical trials. Next is obtaining informed consent. So, informed consent for research participation is an ethical imperative and an opportunity for an individual to exercise autonomy and make an informed decision about research participation based on their understanding of the benefits and risks. Likewise, informed consent is an opportunity for an investigator to educate the would-be participant about research as well as to establish a respectful relationship with them. However, informed consent, both from a subject's perspective and from an investigator's perspective, is for many reasons challenging even under the best of circumstances. In the clinical care setting, although we are asked to sign a consent form for an invasive or surgical procedure, most patients agree hastily without fully understanding it. Because of the trusted relationship with the clinician, the risks of the procedures may be perceived as manageable. Certainly, the benefits are expected to be direct and personal. Unfortunately, as a result of compromised consent procedures in daily life and in typical healthcare settings, and due to most people's inexperience with research, patients and clinical investigators alike may have little context for meaningful informed consent in research setting. Further weakening informed consent for research participation is the phenomenon known as therapeutic misconception. The research participants' often unconscious presumption that care provided in a research study is akin to clinical care, designed to provide therapeutic medical benefit to the participant. Investigators may also be affected by the therapeutic misconception, as it is exemplified when the investigator is over-optimistic that the investigational intervention will bring about health improvements to the participants. So, for justice and equity, we run into the problem of mistrust of research. So, minority groups in the United States have repeatedly experienced unequal and unethical treatment in research, ranging from participation without adequate informed consent to forced or coerced participation in treatments and studies. Concerns of minority groups in the U.S. include unjust distribution of new resources and the potential for genetic enhancements to actually exacerbate disparities. These concerns must be addressed in the enrollment phase of new trials of CRISPR-Cas9 to ensure adequate representation of minority patients and adequate protection to these historically mistreated groups. Then there's underrepresentation in research. A second barrier to equitable participation in research is underrepresentation in genetic databases that inform future research. Without adequate understanding of the range of clinical variants, it will be hard to tailor therapies specifically to minority populations if less is known about their genomic makeup. Consequently, underrepresented minorities will likely miss out on potential gene therapy benefits. And thirdly, racial and ethnic minorities in the U.S. have very disparate health outcomes and access to health care. In this country, socioeconomic status is strongly associated with race and ethnicity, raising concerns that the benefits of gene therapy will prove unavailable to some of the neediest groups for financial reasons. And lastly, CRISPR does have the potential to treat genetic disease despite the ethical concerns behind it. So, research in mice has shown that CRISPR technology could be used to fix the multiple genetic mutations behind the disease. In 2018, a group of researchers in the US have used CRISPR to cut at 12 strategic mutation hotspots covered in the majority of the estimated 3,000 different mutations that cause this muscular disease. And we're talking about muscular dystrophy in this example of this study. But there are also a multitude of other diseases that could be treated, including Huntington's disease, blindness, cancer, blood disorders, AIDS, cystic fibrosis, muscular dystrophy, and COVID-19. Thank you.